Hey, Nathan, it's Marilyn.
Hello, Marilyn. How are you?
Hey, I wanted to give you a heads up that I'm in the car with my son,
Uh-oh.
so I would only like him to learn new science words
Uh,
today. And so,
all right. Thanks for the heads up.
yeah, no.
I've done a fair amount of media, and I haven't actually blown that yet, but I assume it's only a matter of time.
Be hilarious if you like dropped an F bomb on NBC 13 and it went viral. It'd be
Yeah.
pretty
I
funny.
mean, now would be the week. I am tired and over it. So now would be the time. So Tim, I have a gazillion slides of which I'm going to blow through and or skip over for most, but I'm pretty much game for talking about anything the group wants, and I think I have the primary data to back up most of that. There's a couple things that I am particularly interested in talking about, some of that out of self-interest, others just because I think they're important and they come up a lot. And then I had a request to at least provide some of the vaccine data because I know a lot of people are thinking about that. So I'll be more than happy to offer my 2 cents on the vaccines coming up. And then we'll try to see if I can share my screen but still have the chat open to figure out what else people might want to hear about. I'm not hearing you. I'm not sure if anyone else is.
Can you hear me now?
Gotcha.
Yeah, okay. So, no, I think that all sounds fantastic. We'll start at a couple of after. Do you want to see if you can go ahead and take over the screen? I want to make sure you can share your slides before we get started.
Seems to be giving me the offer. Make sure I grab the right one.
Okay, yeah, good.
Does that look promising?
Yeah, I can see it. Yeah, perfect. All right, we'll give it one more minute and then we can get started.
All right, I got the chat open and hopefully it'll stay that way. So I will try to be responsive to that. But at the same time, I'll let you MC it too, Tim, if I miss something or if you think it's appropriate to chime in.
Okay.
And it seems like an hour should be plenty, but there's a lot of stuff in this topic. So I kind of, like I said, I lumped in a bunch of stuff that I'm going to blow through some of it unless people want to hear a little bit more, and then talk primarily about the things that we're using now and why we are, and the other things to keep in mind is kind of my intent.
Okay. Yeah, I think that sounds perfect. So let's go ahead and get started. So I appreciate everybody joining us today. I thought that with our service being responsible for caring for the majority of the COVID patients in the hospital, and with things seemingly changing so rapidly, it would be a great time for us to kind of reset and have a review of all things pertinent to COVID. So I invited Dr. Erdmann to come in, talk to us, and he graciously accepted. So Nathan, I'll let you take it away.
Okay, so as mentioned, Tim left this pretty open-ended, so I apologize in advance if I drift too far from what it is you were hoping to hear about, but I promise I'm happy to kind of redirect, so feel free to chime in if it looks like I missed something that is relevant that you want to hear about. And I will try to do the best I can to read the room here on Zoom about blowing through some other things. So I was going to just kind of review some of the key bits of data and studies that we have in our hands and how they're being leveraged, and maybe think about ways that we could do that slightly differently, and then also talk kind of about best practices. And then, like I mentioned, we'll dig into at least some summary on the vaccines, because I know that's of high interest. Let's see if it'll let me advance. All right, so this is just, I had heard about the virus back in late December, and then we had a ID conference that second week of January, we started reviewing it. So it's kind of funny just to provide context where this was one of these things that was floating around in China as of January 9th. And then by the first week of February, we had sequence and a syndrome. And then shortly thereafter, we had the first couple of compounds that looked to have activity against it. Part of this is because of how fast science can work. Part of it is because we had a construct to work in, and there was actually a fair amount of work that had been done with SARS-1. So there were some things that were known going in that allowed things to move pretty quickly. But all that said is where we are now versus where we were not very long ago, it's worth keeping in perspective. So the other thing I would add to that is anyone who's been on the COVID wards with me knows is that science is a challenge on its own, but doing it in this environment where everything is changing and there's barriers to everything is really, really hard. Mitigating exposure for our teams, trying to limit what we do for PPE, trying to have a grown-up conversation with someone while you're screaming over high flow and the big blowers and all with all that PPE between you. And then, oh, by the way, trying to collect specimens. And then, oh, by the way, trying to have protocols so we can process all the specimens, where to process them, how to do it safely, how to store them, putting a padlock on the -80 that's next door such that it makes biosafety happy. Like, everything is harder. So all of the data that you see, keep that in mind, because it's a Herculean task in any case to conduct a clinical trial, but in this environment, it's 10 times more so. All right, so one of the first things that came out was hydroxychloroquine, and we could talk about it, but briefly, this institution never endorsed it outside of the setting of a clinical trial. And in retrospect, that looks very wise, and that's going to be a bit of a theme that comes up. Another study that came out early was looking at some old drugs that had been identified for SARS-1. And there was some suggestion that there was benefit here, but it was all based upon virologic studies as opposed to actual clinical outcomes. And again, this was largely ignored, also because they didn't use a placebo. So this has just been part of the nature of this, is that you can try to throw a study together and the data has relevance because we're in a vacuum, and yet trying to leverage that data and understand what it truly means is really tough without having kind of a gold standard study. All right, so speaking of bringing into Act 1, so some of you that were on COVID wards early had patients that might have been involved with this. This was the NIH platform that started looking at pilot drugs, the whole concept being that they're going to look at something, make an initial conclusion, then look at the next thing and try to build on it consecutively. So the very first study was a placebo-controlled look at remdesivir. Remdesivir was thought to have, or known to have, in vitro activity against SARS-1, was found to have in vitro activity against SARS-CoV-2, had been given to humans previously in Ebola, and it was a 4-arm study for Ebola, which remdesivir did not win, therefore it did not see the light of day. So when we started giving this drug to humans in February and in March at UAB, it had been given to a total of 500 humans in the history of the earth. So this was pretty fresh and new. That study infused drug on 1,062. There was a change in the last minute. So there's 1,062 people. The people that got remdesivir, they end up leaving the hospital sooner, and they had less complications overall. So that's kind of the key construct, and that is the primary endpoint of the study. So when we start looking into the details of that, everything on the right side over here was good. Everything on this side was not as good. You can see that a lot of these crossed the line, but the key indicators in most populations end up showing benefits. So that was kind of the premise of phase 1. Another way to look at this data, which I think is actually more meaningful in its visual representation, and this blue is good, red is bad, the top bar is remdesivir, the lower bar is placebo. So here you can see that there were more blues, which means more recoveries, and fewer people that were either dead, or red, which means ongoing oxygenation. And this was the low flow oxygen. So this is kind of the primary driver of how we think about remdesivir now, was the 232 people that got remdesivir versus the 203 people that got placebo, and showing that of this group, and showing that those that got remdesivir did better overall. I'll dig into a few more of the nuances of this a little bit later. One of the other things that we've struggled with both as an institution and globally is trying to figure out the subgroup analysis. So this study was specifically powered to look at people that had COVID and a respiratory syndrome. And so that's what these numbers come from. This is the total, and that's where it met its primary endpoint. But we enrolled some people that were on ventilators, and then trying to figure out, well, can we tease out these subgroups? What it looks like is that there's very little benefit for those that don't yet require oxygen, and that there's at least a not small portion of people that are probably too late in the game to respond for mechanical ventilation. That said, what we've learned with this drug, having given it now to I think we're at 870 courses here at UAB, is that the side effect profile is really quite generous. There really aren't too many complications with it. We see about 5 to 10% of people that might have some liver enzyme elevation, and the only renal toxicity is through the vehicle. So we struggle with how best to use this in the ICU setting, particularly for those that collapse and go right into mechanical ventilation. We recently updated our guidelines to give 5 days for those that are on a vent, unless there's a reason to think that they are kind of out of that window. And that's usually where I get to have phone calls with them trying to talk through that. And then here's another way to kind of look at that data. You can see how the on supplemental oxygen, remdesivir clearly separates itself on the recoveries from placebo, whereas it's a whole lot more messy here trying to figure out what did and didn't happen in those that were on ECMO or mechanical ventilation. So this wasn't the only remdesivir study. There was another one that opened in China, and it actually opened before ACT was even going. This was stopped for futility, but not stopped for futility because it wasn't working. There was actually a trend towards some benefit, but futility because that country went on lockdown with tanks and was no longer able to enroll. So very different than our experience here. And then another early study was looking at the comparison between 5 and 10 days. The challenge of interpreting this is that this has value, but it didn't have a placebo arm, so it's hard to tell, especially when you start comparing global studies and the European studies because they were a bit earlier and had more severe early outbreak versus the mortality that was seen in this study. One of the takeaways was, though, that for most patients, there didn't seem to be a meaningful difference between 5 and 10 days. The ACT study was built out at 10 days starting off, and we're still using that just kind of as a framework of reference. But I don't think for the vast majority of patients that 5 days is likely to be sufficient. Where I entertain conversations otherwise is those that have pretty profound immune dysfunction. And in those situations, if we have someone who has persistent or progressing respiratory syndrome in the setting of really severe underlying immune suppression, I would think about extending a course because those clearly aren't our typical hosts. All right, so everything was going along hunky-dory. We had an EUA in place for remdesivir, and then this study came out. And I have a hard stop today at 5 o'clock because I have to go on to the ACT investigator study, but you can only imagine the angst that was on that group when this study came out. And it's been kind of funny and kind of sad to see how contentious the two groups have been with each other. But this study rolled a lot of people, gave them a bunch of drugs that, similar to remdesivir, had been shown to have at least some in vitro or SARS-1 activity, and looked to see if people died less frequently when they got the drugs. So for remdesivir specifically, there are 2,750 people allocated to remdesivir against their control groups. That's a lot of people that got drug. If you remember, the entire ACT study was 1,062. So there's a lot of power in numbers here. And what they showed was not much of anything. Remdesivir didn't help. It didn't change mortality. And so the way this paper was written on its electronic submission, and then what New England Journal ultimately published, was pretty strongly worded to say that remdesivir doesn't work, and this study is looking at mortality, which is more relevant, and therefore remdesivir shouldn't be used. So this is a different study. There's a bunch of things to kind of keep in context. One, it's not placebo controlled, and also it's not blinded. And so there were sites all across the world, and the investigators would have the ability to say that, well, this person we don't think makes sense for these other arms, but we do think it makes sense for this arm. So you could steer them to certain arms. And so there's a huge opportunity for bias there. Another really big difference is that this study did not require confirmation of a positive test. So if someone said that they had tested positive for COVID elsewhere at some previous time, they went on study. And kind of what we understand about remdesivir is that it's pretty time dependent, which is one of the conversations I have with providers pretty frequently in thinking about complicated cases is, does remdesivir make sense? Because is it early enough in this phase of illness? One other part about this is that this was a WHO study. So this was happening in like Iquitos in Peru, where if you go on oxygen, it's because you brought your oxygen tank with you to the hospital. So what we think remdesivir does is slow down the progression of disease. And so if you don't have any ability to get oxygenation support and then you get put on an antiviral, there's not much of a window to give that recovery a chance to catch up. And so interpreting this data, and where I guess the frustration came from is that there was an argument to be made that, well, this is looking at mortality, therefore it's better than the ACT study. And the ACT study said, that's absurd. We are a placebo-controlled gold standard study. We should ignore this study. I think both of them are wrong. I think that the takeaway here is that if you time remdesivir appropriately in the right setting with aggressive supportive measures, it makes a significant difference. And most clearly manifested in how long it takes people to get better, particularly those that are on the cusp of getting worse but haven't gotten there. But for those that remdesivir is not a panacea to everyone who has COVID to keep them from dying. And I think that this study has value in interpreting that. So it's not a singular solution, but I think I'm very comfortable stating that it's an appropriate intervention. And I would certainly want me or my loved ones to be getting it as early as possible once they kind of start having a respiratory presentation. All right, and shortly after the solidarity data became available, the FDA weighed in and officially approved remdesivir for COVID use, and it really based that on the ACT study, which certainly I'm biased, but I think is the appropriate call. So some little context here. We started, the EUA was offered the first week of May. I was frankly just kind of took on the banner of building out a process for that. So we worked with pharmacy to kind of build that approval process. Prior to just about Thanksgiving, I had approved something like 500 courses of remdesivir for the whole institution. We are now at, I think, 870 courses, and the data here on the right, you can see this is our allocations of remdesivir. So this is our early peak back in July when we had a maximum number of, I think, 140 people in-house. And just look at the numbers of what we've experienced here, November into December. So we've had more usage of December than any prior month for the whole pandemic, and it's only halfway through the month. So it's amazing. And then here is how this manifests. This red line is antiviral usage in the hospital, which is usually a negligible expense for the entire hospital. This is all of the money spent on a week-to-week basis for antibacterials, which is usually a huge number for the hospital. And antivirals actually exceeded antibacterial expenditures here in November. And it's going to be, the remdesivir consumption will probably outweigh all antimicrobial usage for the entire hospital. All right, so looking at Eddie's question, the way this is supposed to work is that what I really want to build, when I talk with someone, is that I'm looking to see when was their initial diagnosis, what is their syndrome, and kind of what is their respiratory status now. So if they've already plateaued and are maybe having a long recovery, like because they have underlying disease, we see people with cardiopulmonary disease, with COPD, that they have a long process and they come to us on 4 liters of oxygen, but they've had a syndrome that has been going on for 2 to 3 weeks and they've been stable. I think that there's no utility in remdesivir at that point. But one of the things that I'll talk about at least a little bit coming up is that COVID doesn't follow a strict timeline. While most people will have kind of a peak of their viral syndrome in 7 to 10 days, we know without question that there are other people that don't make a good immune response and that their viral syndrome persists for a long time. One of the takeaways from remdesivir in Act 1 is that if people have an ongoing viremia as determined by a PCR test and are hypoxic, is that antiviral still works, which if you take a step back and understanding what we know about the disease, that is not a foregone conclusion that an antiviral would actually help. So remdesivir is a potent enough drug that even capturing it in a suboptimal way, when they're only presenting for respiratory syndrome, it still has benefit. The right way to use that drug would be to give everyone that has a positive test for remdesivir up front, and I think it would vastly shut down the progression for the huge majority of people. Challenge is we have no referral system to accommodate that. The cost would be extraordinary because the vast majority of people recover before they get to that point. And so we don't really have a comparable way of giving that medicine. So I think if you've been on the code words, you know that if you have 10 people that come in on 2 liters of oxygen, 6, 7, maybe even 8 of them will do fine on their own in a couple, 3 days, will go home, and they would probably do so without any intervention. But a couple, 3 of them are going to become disasters, and it's going to happen very quickly. And we haven't found a good way to differentiate those, which is why all 10 of those people would get remdesivir. But if you have 10 people that come in that are maybe having some cough, but are still compensating and doing okay, either on the outpatient setting or looking okay on the floor, we wouldn't give them remdesivir because most of them are probably going to manage on their own. If we could find out the 1% of those 10 that is not yet hypoxic that is likely to proceed, I would want to give them remdesivir, but we don't have a good way of doing that yet. So hopefully that is at least somewhat informative. So that actually gets to this point, is that we had the viral phase, and if you could provide remdesivir early to all of these people here, I think it would work the vast majority of the time. And not coincidentally, our political people that have gotten themselves infected have all, despite playing down the virus, have all gotten treated with antibodies and antivirals up front. And strangely, they all seem to live despite them checking nearly every box for comorbidities. Whereas the second phase of illness here is the inflammatory side of things. And so what we know is that remdesivir, when we're leveraging it, is in this area. Here it has no benefit, but here, even when the viral portion is waning, it still has value. And this is where we're trying to make sure that everyone we give drug to lands here as best we can. Now that kind of transitions to the second part of things, which is thinking about the host immune side of things. And there's been a bunch of evidence early on that there's a whole lot of IL-6, that IL-1 seems to be related to those that get more sick. We have a lot of TNF-alpha inhibitors that have been explored, corticosteroids, which we'll talk about, and some other ways of kind of shutting down or tamping down the immune system. All should be targeted here. And the question is how best to leverage those therapeutics. So one was IL-6, and IL-6, again, is profoundly elevated, and particularly those that get really sick. And yet targeting IL-6 alone has been tested in a couple, 3 different ways and just hasn't played out. Similarly, there's early evidence on IL-1 inhibition, hasn't worked broadly for those that have even advanced COVID. I think my takeaway from this is that the immune factors driving inflammation and the lung disease are really, really important, but that these pathways are probably too redundant. And either you're going to need to have a hyper-focused group that you're going to select that is really being driven by IL-1, or you're going to need to give multiple factors to kind of shut down the cascade in a couple different ways. And these studies haven't been able to do both of those things. Either they're not restricted enough or they're not able to kind of shut down broadly enough. So I think there may still be value for these interventions, either for COVID or other inflammatory conditions, but that we have to get better at using them at the right time and for the exact right host. So that is not the case for recovery or dexamethasone. So I want to spend some time talking about what we do and what we don't know about dexamethasone. So this is a study, this is one of these pragmatic studies. So this is again where everyone that came in that had COVID, if they wanted to go on study, they were consented, and there really wasn't additional documentation that went with it, right? You have COVID, you're on study, now we just watch to see if you die or not. People went on control group, and it wasn't blinded such that people could know that, well, they don't fit for one of the drugs, so we're gonna go ahead and put them on the control, or they could get randomized, but you could decline your first randomization if you didn't want that and go on to a second tier. So I think those are hugely problematic studies, and yet I think that they're brilliant to be done, because this happened when people were crazy sick, the health systems were barely holding on, and yet they were able to generate meaningful data. And this study no doubt saved lives, and yet it has things you have to take into context. So the takeaway here is that on this one, just for funsies, the left side is better, whereas the right side is worse as compared for dexamethasone. So the overall is that the people that got dexamethasone, they died less frequently than the people that did not get dexamethasone. That is a good thing. For the people that were ventilated, there was a huge benefit of people that got dexamethasone versus those that didn't. And that benefit was also present in those that were on supplemental oxygen. Interestingly, pretty strong signal that there was harm for those that got dexamethasone that did not require oxygen. Digging into the weeds a little bit more though, this is worth emphasizing, at least from my perspective. This is the mortality rate for people that were not getting oxygen. So on this study, the people that were randomized, 13% of them died in the setting of not requiring oxygen when they randomized on study. I'll show you the data for Act 2. There was a 0% mortality rate for people that did not yet require oxygen and getting care. For the dexamethasone, that number went up from 13% to 17%. Again, so that's where this signal towards harm is coming from in the recovery study. For those that were getting oxygen, it ends up being in between. For those that were on mechanical ventilation, their baseline mortality rate was nearly 41% that weren't getting dexamethasone. Our ICU rates, even pre-dexamethasone, were probably around 20, maybe 30%, depending upon exactly how you defined your time to death. And even here, this was an issue because I think they only used 30-day. But I know one of the concerns here too is that it wasn't sure that if we were delaying mortality or if we were actually preventing mortality as well. Again, this study is hugely important, and it's a huge credit to them that they were able to generate this type of data in this clinical setting that they were in. But the data really needs to be interpreted with context. The way I think about dexamethasone is that it is actively bad before someone has a clear respiratory syndrome. So if you are not hypoxic, Dex is not indicated and is actually contraindicated. Dex does not treat COVID. Dex treats inflammation. If you are mechanically ventilated, it clearly has a benefit. I would argue that for those that are on oxygen only, it is by definition a heterogeneous group. I would say that it doesn't help in between. I think it helps some quite a bit, and it doesn't help others at all, which is why I would advocate for being more thoughtful and kind of really evaluating someone before you start Dex, thinking about, do they have inflammatory markers? Do they have risk factors which suggest they may not do particularly well with dexamethasone? Because there are significant side effects for Dex as well. So remember, that is where all of our Dex data is coming from, is that it's this study that it has, again, huge benefits, but also some things that need to be thought about. All right, so those are our highlighted at length. All right, so Before the recovery data came out, the ACT phase or ACT study moved on to its second phase, which was looking at something called baricitinib. So baricitinib is a JAK1/2 inhibitor, which I will be honest, and I had no idea. I'd heard of them, but really had no kind of mental picture as far as what the heck those meant going into it. So why did it get picked? It is an immune modulator. It is already FDA approved, which always makes rolling out a study much more practical. Unlike the IL-6 inhibitors or TNF-alpha inhibitors or IL-1 inhibitors, it actually shuts down a transcription factor that is upstream of a bunch of those. So it shuts down IL-6 higher up and then knocks down a whole collection of redundant cytokines that are like that. It also has been hypothesized to have some antiviral activities in vitro. I don't put a whole lot of weight in that. I think it's more the anti-inflammatory side that drives this. So this study just came out last week. And then the primary endpoint of the study was that people that got placebo, the recoveries happened by 8 days, and those that got baricitinib, they recovered by 7 days, which seems kind of until you think about it a little bit more. So this is all comers. These are people that are getting more refined COVID care because this is the second phase. So this is May into July of last year. They're also all getting remdesivir. So they're already getting what we had at that time was optimal therapy. And then the whole point of the study, the primary endpoint is time to recovery. And so it met the primary endpoint. So the takeaway here is that baricitinib did exactly what it was supposed to do. Here's a way to look at this data. So this is for people that are on oxygen, supplemental oxygen. This is getting baricitinib. This is not getting baricitinib. You can see that there's more blue and less red. So less people dying and more people recovering with baricitinib. But what's interesting is that this is those that are on nasal cannula when they got enrolled. This is those that are on high flow. So this is a much smaller group, but of those that are set up to get more profound respiratory compromise, baricitinib makes a huge difference. And I would argue that the people that are not getting red here are the people that were going to become high flow dependent later. So one of the challenges of this study is, again, it goes back to that if you have 10 people that are sick, 8 of them are probably going to do okay. And that has to be kind of accommodated for in the power of the study, because what you're trying to do is treat the couple people that are going to get worse. And so baricitinib, you can see it really is helpful in this setting, and I suspect that's where most of it's driving from here as well. So baricitinib seemed to have benefits in high flow. I should have put it up here for comparison, but on the mechanical ventilation side, where you see the benefit on high flow, where it skews the highest point towards benefit, that washes out in mechanical ventilation, which my interpretation of that is that it's not broad enough and not potent enough in the setting of those that get to the point of mechanical ventilation, but that it's quite beneficial for those that are hypoxic. For those that were baseline ordinal 4, which means that they had a respiratory syndrome but did not yet have hypoxia, this one didn't have the clear trend towards harm where the error bar barely overlapped 1, but it does drift on that side. So there's very little benefit and maybe some push towards harm. The other takeaway here is that this is a very broad phenotype, right? So that 1 day was not a magical result of statistics. There's clearly trend toward baricitinib providing benefit across all races, all ages, genders, and how long they had disease, whether they had disease for less than 10 days or greater than 10 days. The other part of this study, and this is one of the reasons that remdesivir was pushed forward as well, is that we looked at adverse events. So for those that were getting the drug, baricitinib, which there was some concern that there may be side effects, either through immune suppression and secondary infections, or there's a signal for thrombosis in those that have been on baricitinib long-term for RA. So there was actually less adverse events associated with those getting drug. There were less infections for those that were getting baricitinib, and the numbers are pretty small. You could argue maybe there's a nudge towards having a couple more thrombotic events with baricitinib, but not enough to pop out statistically. So less progression towards badness and less overall adverse events. This is just a breakdown of who is doing what. So this is the overall study, and this is again the 1 day change in overall recovery. And here you can see where that's leveraged. In the hypoxia, you have the 1 day, but then there's this strong signal that comes from those that have a little bit more degree of respiratory symptoms. So those are the primary analyses. But again, I think that's easy to kind of gloss over that, well, 1 day, it probably doesn't mean that much. But in reality, there's a whole bunch of other things that were secondary analyses that popped. So this was significant for 1 day less overall in the hospital, but 8 days difference in those that had more severe illness, faster clinical status improvement, less need for aggressive respiratory support and less duration of support. Overall, 30% overall versus 100% went faster in sicker patients that required high flow. There was a reduction in those requiring intubation. For those that did require intubation, had less time on the vent. And this study was not powered for mortality, but you can see where the value crosses 1 by 1.09. So it ends up just missing statistical significance for mortality benefit as well. And one of the critiques for ACT at large has been that, well, you're not looking at mortality. The challenge is that it's really hard to predict the power analysis for that. So we would have probably been enrolling 3,000 instead of 1,000 people, which means I'd probably just now finishing up that study here in November, December, rather than having the results available to you. So it's a give and take in a very tough environment. All right, so the obvious question now is that, well, you have baricitinib and dexamethasone data that both are showing that they seem to help some people. So which one should we use? The obvious thing to do is, well, compare the two studies or look at for people that were getting baricitinib and dexamethasone. Challenge is it's really difficult to compare these two studies. So I've talked about most of these things. It was pretty well defined who was going on ACT, whereas pretty much anyone could go on recovery. Who these populations were was really profoundly different. Again, you had a mortality rate of 14 to 17% versus a mortality rate of 0% on the ACT population. It was all blind on ACT, where everyone knew who was getting what on recovery. And what we saw in ACT 2 is that the benefits seemed to be driven on those that were hypoxic versus high flow, whereas the strong signal on recovery came from mechanical ventilation. About 1/5 of the phase 2 study of ACT got steroids, but it was clearly biased in who got it. So this is just to show that this is a secondary analysis in the New England Journal paper if you want to read it, but it's hard for me to take away much of it outside of the fact that we just don't know. And this is the differences in mortality that I think are key to thinking about the populations. So the study that we're doing now for ACT and the one that I'm enrolling on right now is looking at these two drugs head to head. So on phase 4, everyone gets remdesivir, full stop. We still use the 10-day course, not because I think there's a huge benefit in 10 days versus 5 days, but it's kind of what we negotiate with Gilead, and it's kind of allows us to compare across the different arms. So that's what we're doing. And then they get 2 blinded products. One of them is a placebo, and then the other is either going to be baricitinib or dexamethasone. They get the Bari for 14 days, which is the same we did for phase 2, and they get the Dex for 10 days, which is the same as it's done for recovery, and the dosing is the same for each study. We're looking a bit longer because we would hypothesize that there might be some long-term outcome issues with those that get Dex, so we're adding on a 2-month time point, which doesn't seem like that big of a deal, but it's a huge pain in my ass because we're going to try to keep track of these people and bring them back in and actually capture data at 2 months. But we think it's important. And we're adding on another primary endpoint, which again changes all of the stats and how many people you need to enroll. But we want to see how many people actually progress towards mechanical ventilation in addition to how quickly people recover. One of the tricks of this study is that it doesn't really make sense to have someone getting dexamethasone for a while and then converting them over to Bari, because we worry that the side effects that you could get from dexamethasone will bleed into the Bari arm, which means my life is difficult now because if you get more than 1 dose of dexamethasone, you're no longer eligible for participating on this study, which goes to A, my bias, but B, my point earlier that if someone doesn't actively need dexamethasone at that instant, it would be great if we thought about it and gave them at least a little bit of time before they at least got their 2nd dose. So thanks to Brian May and Justin Glasgow, because they're working on kind of updating the power plan so that there's at least a little bit of a window for us to evaluate patients. And then I would push people to at least pause for a moment and remember that dexamethasone does not treat COVID. It treats hypoxic failure in the setting of late disease. And so if someone doesn't clearly meet those criteria, there's no urgency in getting them that 1st or 2nd dose of dexamethasone. It does not treat the virus. It treats the late respiratory complications of it. All right, one question about Act 4 is that we have a recommendation for using anti-inflammatories in those that have hypoxia. So is it ethical to withhold steroids? Hard to compare that recovery population back to what we're doing now in current management of COVID based upon the differences in mortality. I'm, again, my bias, but very comfortable that there's evidence that baricitinib is comparable, if not better than dexamethasone in those that are hypoxic, but not requiring mechanical ventilation given the side effect profile and what the overall outcomes look like. And you do not have to spend very long on a COVID ward to see people that are profoundly hyperglycemic and or delirious in the setting of dexamethasone. which is a problem. Not to mention, I've been doing work in the ICU looking at secondary bacterial colonization and infections, and we started seeing signal early on, and we're seeing a ton of signal now in people that have been getting steroids a long time, which is exactly what you'd expect. But for whatever reason, baricitinib had less secondary infections than a placebo arm. So I think that there's potential advantages there as well. For mechanically ventilated patients, as I mentioned earlier, I think that the recovery data is pretty robust, and it would be hard for me to not want to give someone some coverage of steroids if they go to ventilation. So the way the study would work is that we will offer to stop the study drug and transition them over to open label dexamethasone for anyone that would require ventilation. If someone goes on study and they don't look great, giving them dexamethasone as a protocol deviation and makes my life very difficult. But if we get to the point of mechanical ventilation, that's when I think it'd be entirely appropriate, and the protocol allows for that. So that's a conversation I'm happy to have with anyone who's on service and we're talking to one of their patients, or any patient and their family about what that would mean. All right, I was just gonna run through a couple of things that are happening in the background that affect you guys. So very early on, we pulled together a committee that's been trying to evaluate these data as they come out in real time and make recommendations. So when a new study is published, we send out an email to this committee. We try to evaluate in real time. If there's discussion needed, we'll have an ad hoc call. Otherwise, we meet on Monday afternoons. Now every couple of weeks, where in the beginning we were doing this every week. And so these guidelines are what materialize on the UAB1 site. And so this is updated in real time when we make changes to this. If you ever want to join or have a question for that group, we have Justin Glasgow is on the committee, or you're always welcome to reach out to me as well if there's questions or something that you want to bring up. This is the best way I've had to kind of consistently disseminate what the recommendations are. And this group has pharmacists, rheumatologists, infectious disease, critical care docs, hospitalists, ethicists, people from all over the institution trying to interpret the data as best we can and give real world guidelines. Our kind of default has been needing to see pretty clear evidence, otherwise angling towards having people participate in studies. And again, like I mentioned earlier, I think that's served us relatively well. We are not going to be recommending ivermectin. We are not going to be recommending hydroxychloroquine. We're not going to be recommending vitamin C, vitamin D until we see data that's meaningfully beneficial. As otherwise, we'll leave it in the setting of something that we could study, but probably not going to make a recommendation for everyone on. Another thing is that when we started having issues of other studies opening, is that we formed a committee that I lead, looking at a way to be able to prioritize all open studies. So any clinical trials that are going to be recruiting Department of Medicine COVID patients, they submit a protocol to us. We invite them to come talk with us, and we try to figure out how we can make sure that we give the investigators and patients access to the study, but in a way that does so that we don't have kind of a feeding frenzy for every patient that comes in. That's less relevant in the setting of having 20 to 26 admissions on a daily basis. And yet even then, it still is hard because we would have 2 or 3 groups pursuing the same couple patients in some cases. So overall, from our perspective on the investigator side, I think we've been very pleased with how this has provided some benefit and clarity, and hopefully makes your guys' lives less hectic on the provider side. I also know that we are very much not perfect on it. So if there's some feedback that you think needs to be offered, you're always welcome to reach out to me. But specifically because hospitalists are so integral in managing our COVID patients, we have Orlando and Brian who both participate on that, and they were specifically invited to make sure that we represent your guys' interests consistently there. So feel free to offer feedback if it needs to be done. Here are trials that have been open or are open right now. So it is exactly herding cats like you'd expect as far as keeping all these trials running in their own lanes and working with each other and not against each other. But yet overall, we've had very few instances of multiple trials approaching multiple patients or providers at the same time. Not to belabor this point, but we've been working very hard to try to build a repository of samples too. I suspect most of you have run across us in one form or another, collecting urines or salivas or blood. Right, as of today, I think we've enrolled just shy of 1,400 patients from the hospital setting that have provided samples and or access to their health records so that we kind of get a nuanced understanding of what COVID meant to them. I have over 500 sets of PBMCs here down the hall, hundreds of sets of saline and urine. I think that this is, particularly as we roll out vaccines and the whole pandemic shifts again, this is going to be a pretty crucial resource for investigators looking to try to study this. So thank you to you guys who tolerate it, and particularly the clinical staff who helped make this happen in a challenging environment. All right, so I was going to transition over to vaccines. Already talked to you guys for a long time. Is there other questions you wanted to before I kind of jumped into this at least briefly? That was the traditional count to 5. So I'm going to run in through some vaccine data quickly and then see what it is you guys might want to hear about, okay? My intent wasn't to go into the details of this, but just kind of provide some highlights of it. So there are a whole lot of vaccines that are being explored for SARS-CoV-2. This has been the case almost immediately. Even if everything works perfectly, which is already officially not the case because there's a couple of the vaccines that have been moving towards phase 3 that aren't going to make it there or at least going to be delayed. But even if every single one came to bear, we still have a long ways to go to actually meet the requirements that we need globally for vaccine. We're going to do just fine in the United States, at least in getting people that want the vaccine, access to the vaccine over the next year. Similarly, that'll be the case in Europe. But our ability to kind of vaccinate all at-risk populations globally is going to require a tremendous effort, and the handful of products that we have available so far are not going to do it. So there's reason that even though these first couple are working, that these other ones need to be done. That the cost analysis for the mRNA vaccine platforms are not nearly as favorable as some as the others. All right, so speaking of, these mRNA vaccines are absolutely fascinating. They are not new. Like, this concept wasn't developed in response to COVID, but they hadn't actually been given a platform to be able to be used. Vaccines are a interesting, complicated, and rather disheartening market to think about. Like, you don't make a vaccine because it's going to help people. You make a vaccine because it's going to make money. And then figuring out what vaccine is going to make money for who and how you're going to do it leads to some very interesting calculations. What's been fascinating about COVID is that so much of the money calculations have been thrown out the window. So generally, vaccine development is extraordinarily expensive. It still is here, but it's also generally extremely laborious and long term because the investment is so crucial for each individual step. So a typical vaccine product, a fast product, and our prior record was 7 years. So what happened is it takes a huge amount of money to develop into this phase. And then what happens is everything needs to be complete with this phase. Everyone needs to have a market analysis, and everyone needs to agree that, yes, this still makes sense to proceed to step 2. And then you start over again. Then you put in all the money again, and then you generate the whole thing, you evaluate all that data, and then you decide, does it make sense? And then you invest in step 3. And there's about 30 steps before you finally get to a broadly available vaccine. For COVID, the same steps have all been in place the whole time, but the solution was to throw money at it. And so every step was resourced straight away. So that as soon as step 1 was done, there was already work going in because the resources had already been invested in step 2, 3, 4, 5. And so the timeline hasn't been compromised. It's just been done very, very efficiently because there was a pile of money put behind it to make it happen. The other thing, which is worth keeping in mind, is that this was the earliest possible time to get signal back on vaccines. And 2 very, very important and unpredictable things happened. 1, the vaccines are extraordinary. The level of efficacy and prevention is ridiculous, like well beyond anything we could have expected. And 2, far less good news, our degree of our ability to keep transmission in the community in check and not have people get infected like crazy has been awful. So we have had really, really good vaccines that are being tested in people that are getting exposed to COVID all the time, which that allowed the statistics to drive things forward very, very quickly. So this was literally almost to the day was the fastest we could have gotten results back on the vaccines. So if anyone ever grumbles or conspiracy theories of why the results came back when they did, the only reason they came back as early as they did was because of how many cases happened on the placebo arms, how quickly. All right, so this is the summary data from Pfizer. And remember, Pfizer is the one that's rolling out first. This is the one that has the cold chain issues. Interestingly, it is not known that it's required to be kept at that temperature. It's just that we haven't validated that it be kept at a lower temperature. So it may be that in the not so distant future, we actually don't have the same rigor of control for keeping this product at that -80 temperature. But because it hasn't been validated yet, again, going back to kind of all the research and development stages that have been pressed, it is going to be at least in an early phase. And the only reason we're going to be getting vaccine in the near term is because back in March, when this product was first developed, they started the manufacturing process straight away. They were banking on the fact that it was going to have efficacy and been building out the vaccine manufacturing line straight away. So this is one of the most beautiful charts we've seen in medicine, perhaps ever. So you get your flu vaccine, and then my experience in medical school is I got my flu vaccine on a Friday morning, and I went and did my preceptor clinic Friday afternoon, and I walked in on a lady who was coming in with some fever and headache she was a FedEx driver, I remember her in a uniform. Walk in, and like 3 seconds before I can even say hello, she hurls in the trash can next to her, and she had influenza A. And so I immediately had to go on Tamiflu because even though I was vaccinated, the way the vaccine works, it requires a ramping up of the immune response. So long way of saying that this is not anything other than the fact that it takes time for the immune response to recognize the vaccine product or the antigen and make a meaningful response. And then immediately at this 10 to 14 day window, right when you start making a meaningful antibody, the placebo separates from the vaccine arm immediately. And the number of failures are amazingly small. The people that have gotten infected have not had severe disease. And although this is very, very early, the durability seems to be holding for right now. All of those things were in question. It wasn't clear how efficacious they would be. I was hoping for a 60 to 70%. I mean, that was kind of the line coming from Fauci on down from the NIH. And yet they've been profoundly effective. The durability remains a question. We have data that we've published, and a lot of others have shown that natural coronavirus infection does not make a fantastic immune response, probably because of how much inflammation it causes, and it kind of short circuits an effective, efficient immune response. We don't deal with that in the vaccine. The vaccine is just the antigen and turns on the immune system like it's supposed to. The immune system turns back off, and you make a good response to it. The few side effects we have seen are just a consequence of turning on the immune system, which anytime you do that, there's some symptoms that go along with it, and every once in a while there's the potential to have a more serious side effect. Like for instance, we know that Guillain-Barré is associated with flu vaccination, which seems like a bad thing, except even if you're at risk for Guillain-Barré, the recommendation would be to still get flu vaccination because you're more likely to get Guillain-Barré secondary to getting the flu and having all the immune activation from the flu than you are from the vaccine. So it's just the risk we play having immune systems that anytime you turn on, there's a possibility it may not turn off quite the right way. This is a summary of the side effects associated with the vaccine. The takeaway from these is that it is pretty consistently immunogenic, and it is more than likely that you will notice that you got vaccinated. So there is a pretty clear signal that there is pain, that there is a fair amount of myalgias that go along with it, and some headache. And yet, despite the kind of routineness of that degree of side effects, there are amazingly few serious side effects. So vaccine, you may very well not have a great day the next day. So you may be a little achy or a little tired, but the likelihood of having a more severe side effect is extremely low. We've seen almost nothing, and no like necrosis or abscesses, sterile abscesses, some of the other stuff we've seen with vaccines in the past. It is, again, you may notice that you've gotten a vaccine, but there really isn't much more to it, which is great. Moderna is the second product that just kind of has jumped through the FDA hoops. This one has a little bit more forgiving cold chain, and the data looks amazingly similar. Again, nothing happening at day 10, and then the curves quickly separate. This data is particularly reassuring because I didn't dig into it, but the Pfizer data only had 10% of kind of underrepresented minorities, whereas Moderna is able to enroll almost 30%. And the vaccine response is essentially uniform across all of the arms. So everyone seems to be responding in a relatively similar way. Old, young, different demographics, everything seems to work. And again, that was not a foregone conclusion. Again, pretty darn immunogenic vaccine. So good chance you're going to realize that something's up. There have been a couple of cases of Bell's palsy, which again, if it's related to anything, it'd be non-specific immune activation as opposed to really something that seems to be a specific signal to the vaccine. Some headache, some aches, quite a bit of kind of yucky tiredness, and the occasional fever or chills to go along with it. For what it's worth, I have received my email, responded to my email, and look forward to hearing from the powers that be so I can get vaccinated without reservation, and would recommend anyone with just about, I mean, this isn't a live virus, so I just have very, very few reservations for people getting vaccinated. It's we're amazingly fortunate to have it this quickly. And again, it took a constellation of things to happen for us to be at this point. All right, I put in a whole lot of people that I've been working with tirelessly for the last year that have been extremely important to it. And I could frankly put just about every one of you guys. So thank you for plowing through and being available and cooperative with me too. And I've been chasing around for all the various study things and samples and everything whatnot. So I appreciate everyone's efforts. Hopefully that was helpful and not too much just blawing on, but I would happily entertain whatever questions you might have.
That was great. Thanks, Nathan. I really appreciate that. I had a question that wasn't directly related to clinical practice, but more out of curiosity. I think there were a couple of papers back in the spring or early summer in the New England Journal of Medicine and Lancet that showed that there was benefit to hydroxychloroquine, and they later had to redact those papers. And things like that have only made it harder when dealing with the public who have, general mistrust. What leads to that kind of paper being published in some of the most prestigious journals around?
So the hydroxychloroquine was interesting all around because I think people lowered their bars a bit trying to give the benefit of the doubt. The studies that were published early on, there was a bit of a feeding frenzy from the journals of knowing that it was going to be high vis. And the journals, I think, approached it with the opinion that as long as we're presenting the data. We're not presenting a value judgment, we're presenting the data. And what the data was case controls, non-placebo controlled. And it is what it is, right? It's not wrong data, it's just it's very easy to overinterpret. So the very first data on hydroxychloroquine was coming from a cohort of 27 patients. And there were 5 patients that were dropped that had all gotten hydroxychloroquine and had all not done well. And I don't want to imply that there was anything nefarious about that. It's just that it was impossible to make a meaningful conclusion. Again, our institution read those papers, discussed them, and really felt that there was nothing there that was going to be benefiting our patients now. And we look forward to seeing more data, whereas others kind of latched onto that and overinterpreted what was present. That is always the case in science, right? Like that's not a new phenomenon. It was just particularly acute in this setting where everyone was grasping for bits of information, and there were a lot of experts that weren't so expert in a variety of platforms. Not to argue that me or anyone here or any one particular person was infallible or worthless, but there was a lot of noise and things weren't always interpreted the most appropriate way. Other questions? I'm again, my bias here, I'm certainly interested if there's questions, feedback, reservations about Act 4. I'm pretty motivated because I think it's a really interesting study. I know a lot of us aren't really big steroid fans, and yet somehow we're all throwing steroids around like crazy. And I think getting more nuanced data, particularly randomized data for how they work would be informative. But I know there's also a comfort level with people giving steroids too. So would be happy to entertain that if there's specific questions.
Hey, Nate. So
hey,
we have been.
That's a fine mustache, sir.
We have been successful with mRNA vaccinations with COVID. How is it so different than like HIV or Ebola or other stuff? Is it just because of the spike protein, or what's so different for not being successful?
So COVID is a coronavirus, and coronaviruses don't make fantastic immunity because of complications of inflammation. But the immune system is able to see and chase the antigen and the virus itself without a whole lot of trouble. The problem with making a vaccine against HIV, against TB, against malaria is that by definition, the pathogen doesn't make an effective immune response. So if you're going to make a vaccine response to HIV, what immune response should it look like? And we honestly don't know. We have pretty darn good ideas now based on work that's been done for the last 10 years. But early on, the assumption was feed the immune system antigen. The immune system will make antibody to that antigen, which it does. That should help prevent infection. It doesn't work that way because the virus changes and the way the virus works. Similarly, TB and malaria, the way the antigen interacts with the immune system doesn't make a reliable immune response, which means just triggering an immune response is not sufficient. So, like, I never worried about Ebola as a pathogen to make a vaccine to because it's a relatively straightforward pathogen. It's a nasty disease to get, but it's a relatively straightforward infection. Coronavirus is more along those lines, although that was less clear when this all started. So I was certainly not while I championed for vaccines, I wasn't relying upon a 100% vaccine response straight out the gate. I guess the one other thing which I think is worth highlighting for the mRNA vaccines is that we make protein vaccines with some regularity, but it's hard to make those proteins. It's hard to manufacture those proteins and to get the proteins to look exactly like the immune system would normally see them. And the mRNA ends up being faster. And then the way the body folds the mRNA product into protein is exactly how it's supposed to be, which is part of the reason they're working so well, it seems. Hopefully that was helpful.
Yeah. And Nathan, there's a question about convalescent plasma. Are there current guidelines for use with that?
Yeah, I had a slide in there. I must have chopped it out when I was throwing things together. My apologies. I'm glad you brought it up. All right, so what we know about convalescent plasma and monoclonal antibodies and antibody therapy at large. It looks like antibody therapies are very, very helpful if a host, an infected person, does not yet have antibody, all right? The problem is that most people, by the time they have respiratory symptoms, they had a lag that they are set up to have more severe disease, but many of them already have antibody responses. And by the time to that point, they're more at risk for kind of immune-mediated trouble. So sorting out exactly the timing there in the severe disease is more difficult, but antibody therapies likely are very beneficial early. The problem there is scale, logistics, and cost, because if you give an antibody therapy to everyone that gets infected, it would likely work. But there's no way for us to do that at scale right now. But for those that are already hospitalized, that are already having respiratory compromise, we know now that the Lilly monoclonal antibodies seem to have no benefit. Convalescent plasma seem to have some signal for harm, and certain at-risk populations didn't seem to have really a benefit. And the other thing is, again, my bias, but ACT rolled out these randomized placebo-controlled studies right away, and the antibody studies have really struggled to kind of make that happen in parallel as well. So I think that's also partially because of, there's a lot of things that go into that. But the takeaway seems to be, and this is reflected in where the investment is going, is that there's very little role for antibody in the setting of relatively advanced disease. And there's opportunities to have really profound impact early on, which is why we have an EUA program going on right now with those same antibodies early. So people that are now feeling sick and are getting referred quickly, there it probably does have a role. But the logistical challenges there are tough, and we just can't roll that out at scale yet. Which goes back to the other question too. If you have 100 people that are infected, 80 of those people are going to do fine. If we could find those 20 people that are set up for more severe disease, that's where we'd like to leverage those products. But there's still a ton of cost, and the infrastructure to get those people identified, tested, referred, and infused all in a very short amount of time is a tough ask, particularly for our at-risk populations.
I got a question.
Shoot.
Real quick, sorry. Mute our TV in here. Our current power plan, you know, it's got the LDH, ferritin, CRP, of course, the dimer, all that kind of good stuff. And, you know, in terms of trying to understand, you know, those patients in terms of risk, say you have a patient comes in, they're not hypoxic, you order that enhanced respiratory power plan, you get some numbers back on them and their inflammatory markers are really high. Would that be a good patient to go ahead and, would you still hold DEX? Like say those numbers, those inflammatory markers are really high, would you still hold DEX on that patient if they're not requiring oxygen?
Good question. So if they are not requiring oxygen, it's going to depend. If I'm convinced that they're headed that direction and they have really high inflammatory markers, I'd think about it. But I would still really be thinking critically about if I want to wait and determine. Now, if they're not requiring oxygen at rest, but they're clearly desatting, they have a lot of fluffiness on their X-ray. Again, those things will start kind of tipping the balance towards being more aggressive with with treating with DEX. But more often than not, people are being started on DEX before even those inflammatory markers result, which I think is misguided. Until you realize that someone has a ferritin that's well over 400 and maybe pushing 1,000, until you have a CRP that's well over 80 and maybe pushing 200, you really don't know. And without those inflammatory markers, and I don't want to overstate it either, because it's not that we don't have fantastic uniform biomarkers like this person is inflamed, this person is not. Ferritin is an approximation, CRP is an approximation. But when you put the whole clinical picture together, someone who has robust infiltrates, a ferritin of 712, a CRP of 126, they look awful. And they have kind of ongoing viremic syndrome, like they're achy and they're short of breath and they're coughing. I want to give that person everything, whether or not they're on 2 liters or satting on room air at 92% or on high flow. Like, they need treatment. Whereas the person who looks pretty good, their CRP is 36, their ferritin is 108, and they appear okay, but they came into the EDAR and they got thrown on 2 or 3 liters. That person I would sit on and kind of try to let them declare what they do and don't have going on. Because it's more than likely, most of those people, they're probably going to leave in the next day. And if they do get worse, we kind of have time to calibrate what is and isn't going on with them. So there's no one marker that I would be comfortable with saying that is my decision tree. But having looked at hundreds of these folks, you can kind of start lumping people that look like they're more primed for badness versus this person looks pretty good and there's really not a whole lot of danger coming from their initial lab panel.
Awesome. That's super helpful. Thank you.
Yeah, I wish it was a little bit more clean than that. I think that there's the potential for having personalized medicine to know that here's a few key factors in certain demographics for people that are really, really high risk, but we just don't have that degree of nuance yet. So it kind of takes lumping a handful of markers and getting a kind of collective gestalt on it. But if you've been on COVID wards for more than once or twice, you can walk in a room, sick, I'm nervous about you. You look fine, we just need to give you a day or two. Day or two person almost certainly doesn't need Dex, and you're more likely to cause them harm than benefit in a lot of cases. The sick person, and then you look at some labs and everything is consistent, that's someone who whether or not they go on study doesn't matter as much as just treat in the best way you can.
All right, Nathan, I know you got a 5 o'clock, so we'll let you go, but thank you very, very much for doing this. And thank you for all the work you've done the last 10 months. We all appreciate it.
You guys have been good partners. Everyone's busting their butts, so I sincerely appreciate it.
Great.
Don't be shy if there's something I can help with, okay? Have a good night.